Okay, good morning from my side as well. Just a quick warning, we should have probably put David Josephi's Just 101 session before our session. So there's a lot of research data ahead. I tried to make it easy for you to understand, but well, I guess we could take some questions in between, or we could do it at the end, or we are around later if you have any particular questions that you want to ask. So, as Brian said, this is the first project that we have put together as a group where everyone of the group has actually contributed something. Also, again, we have been collaborating before, but these were more single projects where parts of the group have collaborated. So this is really a whole team effort. So what we have done, and this is part of the DJ whole genome effort, is a lot of high-throughput technologies, sequencing, whole genome sequencing of many GISTs, looking at gene expression on the RNA level, and then put together everything and reporting what we found. What we did find, we did find a lot of things. Of course, the main thing that is important for you is that there are genetic events in GIST that can help us risk stratify tumors, especially when the question stands for adjuvant therapy. And the next big thing is that potentially from these findings, we get more cues for additional therapeutic approaches. So the really great thing, or the big news, is that we found alterations in genes that take part in regulating something that we call the cell division cycle. They can have something wrong in one way or the other, and that tells us something about the risk of that particular tumor to get worse. What is the cell division cycle? That is a cellular machinery that regulates how fast or how often a cell divides, so a cell duplicates. So what we found is when we looked at the genome of a whole bunch of GISTs and sequenced them and looked at their cell division cycle regulating genes, some of them had something wrong and some didn't. And when we separated those out and looked at how the patients were faring with respect to progression-free survival, that is listed here, then we had two groups, the group with a blue bar, blue line, and the group with a green line. The blue line over time did really well. Those patients that did not have anything wrong with those genes, they lived out a long time before having any disease progression, whereas you can see the group of patients that had something wrong in their tumor that in those genes had progressed more quickly. So this adds a piece to the puzzle of how GISTs develop over time, from a small microgist to a proliferating GIST that gives the patient some problems and needs to be taken out by surgery, to an invasive metastatic GIST that is overtly malignant. So we know that all GIST, even the smallest ones, have mutations in the KIT or PDGFR alpha genes. We don't exactly know what needs to happen to have a small GIST get into something that is more aggressive. However, we do know it from a more broader standpoint. Something is happening at the chromosomal level. Some chromosomes get lost. And further along the way, we now find that something's wrong on the genomic level in those cell cycle regulating genes is the next thing that happens on the way to a metastatic GIST. The question is now for us, what are the missing pieces in this timeline, and find out some of those other chromosomal alterations. But Jonathan's lab has recently found one thing that is really very important in making a GIST really aggressive and metastatic. It's called a gene called dystrophin, and I'll briefly touch on that later. So Brian has mentioned the sort of single projects of the life-proof pathway to the cure, and has gone through this diagram, which has a lot of things that are on our plate. I'm not going to talk about all of these, but what these research priorities boil down to are essentially a few things. And these are the things that our research team focuses on. It's looking at the basic biology of GIST and developing new model systems that can help us to test new therapies. We look at predictive and or prognostic biomarkers that will help us in patient management. Of course, the main goal is to find new therapeutic targets and new therapies. So this is how the remainder of the talk is going to be structured. I'll show some vignettes, some research results in each of those categories. So let me start with what the group has been doing in the category of basic biology and new GIST model systems. So here I will start with Tamas Ordog, who is the expert on the precursor cell of GIST, interstitial cells of Cajal. His hypothesis currently in the lab is that something that we call the GIST stem cells, which are even precursors of the interstitial cells of Cajal, or, well, early cells that develop into interstitial cells of Cajal, that they may, when they contribute to GIST tumor formation, that they can contribute to therapy resistant in GIST. So this is what he's working on, and some observations that he has made. He found that KIT mutations in those ICC stem cells increase the numbers of those ICC stem cells, and he also found that the ICC stem cells are insensitive to Gleevec treatment. When he transplants those cells into a nude mouse, you can see that those mice develop tumors. Excuse me. And when a pathologist like Brian looks at those tumors under the microscope, they look like a GIST. Interestingly, thank you. Interestingly, these tumors express not so high numbers, not so high levels of KIT, since these are developed from the stem cells. What this kind of reminded us of, though, is that this resembles a number of GIST tumors that had been under imatinib therapy and maybe grown to be resistant to imatinib therapy over time, which also do not express much KIT anymore. So the main goal of his research is to maybe restore, to restore KIT levels in GIST cells to make them amenable to therapy again. So now coming to another basic biology project in Jonathan's lab. I mentioned the dystrophin gene already. What is this? It's a gene that is actually known very well from research in skeletal muscle disorders. Its function is disturbed in muscular dystrophy disease or disorders, and it sits sort of in the muscle cell and links it to the outside and makes the muscle cell function. What Jonathan's lab found, though, is that this molecule is expressed in GIST, as you can see here. This is a GIST that has been stained for it. All the brown means that this protein dystrophin is expressed, but only in small localized GISTs. When you look at aggressive metastatic GIST, you don't have that brown staining anymore. Dystrophin is not expressed anymore. And through functional studies, like really important functional studies in Jonathan's lab, he found that inactivation of dystrophin in GIST is actually linked to metastatic behavior of GIST. So we now have another puzzle, a piece of the puzzle of GIST development into an aggressive tumor on one side. However, since there's much interest in how to target dystrophin in muscular disorders, where we try to restore dystrophin expression in some way or the other, doing the same thing in GIST would also help to maybe dampen the pro-metastatic effect and that means that any therapy that people in the dystrophin field find for muscular disorders may also be good therapies for GIST. So it can potentially suppress GIST metastasis. So let me get to speaking of aggressive GIST and how we treat it and the problem of GIST resistance. Let me get to our first model that was developed in Sebastian Bauer's lab. This is a really cool model, as you can see already. Two nice pictures with colorful cells. He developed a mouse model of GIST that more resembles what is actually happening in a patient, but at the same time allows us to evaluate therapies that we are looking at, for example, really quickly, and at the same time trying to evaluate the mechanisms of resistance better and easier. So what he did is he developed in the lab GIST cells that are colored. These have different mutations, and each mutation is represented by a different color. So all the green cells that you can see in here have a imatinib-sensitive KIT mutation. All the other colored cells have different mutations, but they're all Gleevec resistance mutations. So all the little red or blue or even sort of gray spots that you see in here have a different mutation that cannot be treated with Gleevec. So he puts those cells in a mixture into a mouse. These cells grow a tumor, as you can see up here. Then he treats the mouse with Gleevec and see how the ratio of the cells that are either sensitive to Gleevec or resistant to Gleevec, how that changes. And as you can see on the right side over here, there's not that many green cells that you can see, but a lot more red and blue and gray cells. So he has killed all the green cells, but now the other colored cells with the KIT-resistant mutations, they don't care about Gleevec, so they grow up and can develop a new tumor clone. And this is sort of something that will happen in the patient, and we can even look at the different cells now here under the microscope. So what are we going to do with that model? We can look at whether we can prevent resistance by changing schedule, dosing, drug sequencing, or drug combination, and find really easily which cells are affected by which drugs and how we are doing with that. And then we can also study more mechanistically resistance in a way that when does resistance to drugs start, and when should we even start treating that? So the next project that I'm going to touch on is our KIT wild-type GIST, or pediatric-type GISTs. This is one of the main focuses in Oregon. Chris Corliss and Mike Heinrich are depicted here. Chris is a world expert in, gene sequencing, and so what he, together with Mike, is doing is they are taking a group, a larger number, a growing number of KIT wild-type, pediatric-type tumors, and sequence them, analyze their genome, and try to find out what's wrong with those tumors to find new therapeutic targets. And what they have developed is to make this process a little quicker than maybe looking at all genes in the human genome. They have developed a panel of 23 genes that are sort of hotspot genes that the research community is interested in looking at, and where mutations in GIST have been found before. This is complemented by a model that Mike is working on because, as many of you know who are interested in pediatric wild-type GIST, we unfortunately do not have any good models, speaking of cell lines or mouse models, that we can actually even study the biology of these tumors very well, and even not speaking of testing new therapies in a fairly quick manner. So what Mike has turned to is a yeast model, which may seem strange. It's a regular brewer's yeast, what is used to make beer, and he knocks out the SDH gene in those yeast cells. So the SDH gene is something that is mutated or at least altered in a different way in a proportion of pediatric type GIST. So he produces yeast that do not have that gene, and now these yeast strains can be looked at and tested for their biologic behavior and potentially tested for new therapies. Why does this work in yeast? Because yeast are not humans, obviously. Many of the genes and intracellular pathways are actually the same between yeast and humans. So many of the much of our understanding in genetics as a whole actually started in yeast studies and yeast genetics, because yeast is such a great model system for these types of studies. You can alter their genome really easily and then do functional studies on them. In the same vein, another KIT wild-type model is being produced, and this is really hot off the press, and we are really glad that eventually something has worked. Sebastian Bauer was able to take a patient tumor from, a GIST tumor from an NF1 patient. These GISTs do not have KIT mutations normally. He put that into a mouse and that tumor grew. So we have something growing in a mouse. We hope that that keeps going. And what he's trying to do right now is to take that tumor out and put it in a plastic dish and try to grow it. So we have nice upcoming models for KIT wild-type, pediatric type just coming up. Our next topic is going to be what the group has been doing about predictive and prognostic biomarkers. What I'm going to present is a large study that Maria Dabietz-Richter has put together and published really recently. She is looking at whether the KIT mutations that are present in a tumor, in a GIST, and you all know we can have several different kinds of KIT mutations that affect different proportions of the KIT gene, if they have a prognostic effect. So her research group, in collaboration with many centers in Europe, have looked at thousands of GISTs, looked at their genetic profile, and then looked at disease-free survival of those patients and tried to stratify whether the mutation makes a difference with respect to prognosis. And what she found is that, yes, there's actually a certain KIT mutation in KIT exon 11 that is an adverse prognostic factor. This is represented by the blue line here. So again, we are looking at disease-free survival. This is depicted on here and over time. So what you can see here, that this blue line goes steeply downwards, which means that patients in this group had a not so long disease-free survival. So after only a few months, they had recurrence of their disease. However, when you compare those tumors with that blue mutation to other tumors with a different KIT mutation, either in, exon 9 mutation, for example, or other exon 11 mutations, or even PDGFR alpha mutations, they are a lot different because they live longer without recurrence of the disease. Interestingly, however, this is only true when you look at gastric GIST. What you can see here is the gap that I explained to you between those two groups in gastric GIST. However, this does not happen if you look at GIST that are arising anywhere else in the gastrointestinal tract. So again, here she compared this specific mutation with a bunch of other mutations that can happen in GIST, including PDGFR alpha mutations. She then only looked at KIT exon 11 mutations, and sort of the same thing happened. The same mutation fell out again as being different than any other KIT exon 11 mutations. And this essentially almost looks the same as what I showed you before. Same result, only found in gastric tumors. So why is this important? This is important because we really need to get a better handle on patient selection, especially when we are looking at which patient would be better off receiving adjuvant Gleevec after tumor surgery. We now look at certain risk stratification schemes that kind of look at the size of a tumor, how fast a tumor is dividing, and this works to some extent, but we need a better differentiation between those tumors and to really tell each and every patient what he or she needs to do. So this will further help in selecting those patients that may need therapy earlier than or adjuvant therapy at all versus those that don't. And this is especially important because that specific mutation that has the adverse effect of progression-free survival, that's actually sensitive to Gleevec therapy. So it's sort of an easy fix. We're now getting to the next category, new therapeutic targets, and I should actually have started with the second subcategory in here before, but I haven't had the time to change my slides. So now what we're looking at is what Matt Van der Rijn's group at Stanford is doing, one of his projects. They found a new therapeutic target. It's called ROR2. You don't need to know a lot about that, other than maybe it's a type of molecule that is very similar to KIT or PDGFR alpha. It's also called a receptor tyrosine kinase. It is expressed in GIST, and again, when you look at patient outcome, ROR2 correlates with this. If you have more ROR2 in your GIST, then this leads to more aggressive disease. He then can look at ROR2 in functional studies in the lab. So he put more ROR2, represented by the brown staining here, into GIST cells. And when he looked at how fast those cells grow and if they can maybe produce something that we call a metastatic phenotype, he's seeing, yes, these cells that have more of this ROR2 can invade into other tissues. So that's another important piece of the puzzle. But then he is going one step further and trying to see, well, what happens if we get rid of ROR2? Could that help therapeutically? And yes, when he takes out ROR2 of the equation and looks at mouse models in which he's doing that, the tumors get reduced and they have a lesser metastatic potential. So all in all, ROR2 may be another therapeutic candidate in GIST. The next step will be to find a drug that can target ROR2. So I'm sure he's working on that. So the next project is actually something that goes from understanding basic biologic concepts to developing therapeutic targets in GIST, and maybe you recognize the lady up there. It's happening in my lab, and it started with an observation that many of you have made also, which is Gleevec is a good drug, and it kills a subset of tumors, which you can experience yourself in the tumors shrinking when you take the drug. However, the tumor oftentimes does not go away. It stays there, it doesn't do anything, and you can, many people are actually living with that for a long time, luckily. However, if you would look at these tumors under the microscope, there are still GIST cells in there, and they look alive. So essentially, if you took Gleevec away, those cells would start to grow again. And this is what we have actually been looking at in the lab. We were able to reproduce this in the lab. You can see this on the chart on the right. In this experiment, we treated GIST cells with Gleevec for a while, 3 days. And what we were looking at was how many cells are in the process of cell division. And this goes down from about 30% of cells, that's normal for what we see in tissue culture, to almost zero after those 3 days. However, when we take Gleevec away at this time point, it takes a little bit, but the cells grow back to what they've been before. So we are able to recapitulate what is happening in the patient. So we now want to understand that process because, as I told you, the cells can not only wake up again, but these cells could be a potential reservoir in a patient for resistant clones. So this is understanding two parts of this process. What we did find is that something that we call a protein complex, it's just a clump of molecules working and acting together, essentially depicted here. You don't need to know everything about it, but the name is kind of catchy. The dream complex is involved in putting the cells to sleep. It does not only do that in GIST cells after Gleevec treatment. It's a universal complex in this molecular complex in the cell that is involved in normal sleep processes also. But again, it is happening after Gleevec treatment in GIST cells. So our next question, though, after just finding out that this important complex is involved in the process is, can we maybe target that process therapeutically so that we can push more cells, more GIST cells, into cell death? And one regulatory process of the DREAM complex gave us the opportunity to do that because DREAM complex is regulated by something that we call a kinase. And you hear that name. Kinase reminds you of receptor tyrosine kinase, what KIT is. And kinases are molecules in the cells that can be targeted with drugs fairly easily. So this is what we actually were able to do. There's a drug that targets the DREAM regulatory kinase. And we tried to ask the question, if we use that inhibitor together with Gleevec, can we kill more cells as Gleevec alone? And this is actually the case, and you can see at this experimental look at this experimental figure here. It's kind of easy to understand. IM stands for imatinib. If you don't, and harmine is that other inhibitor that we are using, the sleep inhibitor. So if you don't treat the cells with anything, you don't have any of those black spots here, which stand for a protein that is upregulated when cells die. So no treatment, no cell death. That's what we would expect. If we treat the cells with imatinib, this is the little plus sign here, we see some gray blobs coming up. That means that the cells are dying now after the treatment. However, when we combine imatinib with that sleep inhibitor, you see a huge dark black blob. That means that a lot more cells are dying right now. So this is something that we found fairly recently. We're trying to now put that into more clinical studies, also together with Maria Dabietz-Richter. Well, no, Dabietz-Richter. I just found out about how to pronounce her name. So using that as a new therapeutic target and Brian is actually looking at a different angle of cell sleep, or the technical term for that would be quiescence as well. So that's another sort of angle that started or project that started in one single lab and then branched out to other members of the research team, which makes our team so nice and so very productive. So in essence, we found that drugs work better if the cells can sleep, so now we want to do something about it.